Hello. Thank you for watching today's presentation on overcoming the challenges of working with diabetic mice. My name is Danielle Fontaine. I'm delighted to share this educational information with you to support your efforts to achieve research excellence. If you have any questions about the content of this presentation, please contact us using the information on the final slide of this presentation. Before we get started, I'd like to share with you some information about the Jackson Laboratory. The Jackson Laboratory is a nonprofit biomedical research institution. The mission of the Jackson Laboratory is to discover precise genomic solutions for disease and empower the global biomedical community in our shared quest to improve human health. We accomplish this mission through several aspects of our organization. We have our own research staff with their own private funding means, such as NIH grants, that investigate the genetics and biology of human disease, partly using the laboratory mouse. We provide scientific services and resources to investigators around the world. In addition to our clinical and research services, some of you may already be familiar with, we also have some freely available resources that I'll touch on in more detail shortly. And finally, here at the Jackson Laboratory, we're very committed to education outreach. In addition to webinars such as this one you're participating in today, we also sponsor traditional conferences and hands-on workshops, as well as educate students of all ages through courses, internships, and other programs varying from high school internships to postdoctoral assistantships. The Jackson Laboratory is the world's largest repository for mouse models of disease. We are NIH funded to import and cryopreserve as many mouse strains as possible to ensure their availability for all time. Among the more than 11,000 strains we distribute at both our Bar Harbor, Ellsworth, and Sacramento facilities, over 1,500 strains are maintained as live colonies. JAX mice are the best characterized and referenced mice in published research, and much of this can be attributed to rigorous animal health standards and unsurpassed genetic quality. And what many investigators may not realize is that the use of our well-characterized and heavily referenced common inbred strains, such as the C57 Black 6J or bulb CJ mice, supports the development, collection, and distribution of specialty mutant mouse models, as well as provides funding for our freely available resources. So some of our freely available resources include our JAX mice database. For any of you who have looked for available mice on our website before, you may have ended up on our JAX mice database, which has data sheets for every strain in our repository. While your initial goal may have been to see if the colony we have is live or the price of the mice, if you spend a bit more time on the data sheet, you'll see we have tons of other useful information, such as the genotyping protocols that we use here at JAX, phenotype information associated with that strain, how it was developed, genetics associated, and even a list of helpful references that utilize that strain. The Mouse Genome Informatics Database is a great resource that includes information not only about mice from JAX, but also mice from other repositories and other investigators. I like to think of this database as our one-stop shop for all things mouse genetics. It's a useful way to search for strains that carry a particular allele, and if they're available through any public repository, JAX or otherwise, you can link out to those resources on the allele detail page. The Mouse Phenome Database is a collaborative collection of baseline phenotype data for some of the more popularly used strains. And again, all of these are free online resources available to help you with your research needs. So in today's presentation, we will be discussing the many possible characteristics of diabetes that can be modeled in the mouse and how this might require special care. We will also go through handling and housing strategies to improve animal welfare and obtain more consistent experimental results. So very quickly, I'd like to just go through the two main types of diabetes people are often modeling in mice. To start, insulin is a hormone produced in the pancreatic beta cells and activates the uptake of glucose into organs throughout the body to use as fuel. Both type 1 and type 2 diabetes have beta cell dysfunction, though in very different ways. Type 1 diabetes is an autoimmune disorder where the body attacks and destroys these insulin-producing beta cells, resulting in little to no insulin production. This results in high blood glucose levels, which has several negative impacts on the body, such as those that are listed here. Individuals with type 1 diabetes regulate their blood glucose by administering insulin injections to supplement the body's lack of production. Type 2 diabetes involves normal pancreatic function, at least in the early stages. This disease is correlated closely with obesity and likely has multiple underlying genetic and environmental factors that contribute to disease progression. Here, while there is normal insulin production, the peripheral tissues of the body are resistant to glucose stimulation. This glucose stays in the bloodstream, resulting in hyperglycemia. The insulin resistance results in the pancreas struggling to produce more insulin, eventually leading to beta cell failure. Therapies for both types of diabetes are important to improving patient outcomes and are often modeled in mice. However, there is no perfect model for human diabetes in the mouse population. For both type 1 and type 2 diabetes, we don't yet fully understand the mechanisms behind their progression. Using a mouse to model characteristics of diabetes can help researchers understand underlying genetic factors that might lead to disease progression, help model high blood glucose or insulin resistance. There are several popular mouse models in the published literature, but all models are not created equal. Different models will recapitulate different aspects of each disease, so it's important to understand what each model can and cannot provide for your research. Here you can see several of the more popular models. As you can see, we have several models that mimic obesity. We also have models of autoimmune beta cell destruction, such as this NOD mouse model here. They all look very different, and they do model different aspects of this disease. Before you begin your experiment, it'll be important to know which mouse model will best suit your research. So the first challenge to working with diabetic mouse models is, what characteristic are you looking to model? Is hyperglycemia all that matters, or do you need a model of cardiovascular disease associated with diabetes? One common issue with human patients is delayed wound healing, which not all mouse models will repeat. It's important to have a clear understanding about the phenotypes you're interested in modeling before starting your studies to ensure that you pick the model that can best answer your research question. Once you've picked an appropriate model, the next challenge is designing your experiment. As with any disease models, many of the strains available do not have a 100% penetrant diabetes phenotype. Some models may have a higher disease incidence in one sex or at a particular age. Based on your experimental treatment, there may be non-responders as well. All of this should factor into how to properly size your cohorts to obtain useful data. Speaking of data, how are you collecting yours? Is there a specific time of day that's best to collect blood glucose or body weight samples? Control selection is also a big consideration to have sorted before you begin. What specifically are you controlling for? Is it a diet, a genetic background, or a mutation? We always recommend performing in-depth literature searches of your field to determine what is considered an acceptable control for both the mouse line as well as any experimental assays you may be running. There are also many environmental factors that can affect phenotypes, especially when we discuss models of type 1 diabetes. We'll go through a few examples of this later in the talk. Often a best practice to ensure accurate conclusions is to make sure the environment is as similar as possible between your experimental and control groups, housing them in the same facilities and rooms if possible. One example of the importance of knowing the characteristics of your strain before you begin your experiment is the NOD mouse, or non-obese diabetic mouse model. This is a well-known model of type 1 diabetes-like phenotype. These mice have immune infiltration and destruction of the pancreatic beta cells. However, as you can see from this graph, disease onset is variable and also sex-dependent. About 80% of females and 40% of males are diabetic by 30 weeks of age, and we can take 3 major experimental planning details from this. The first, not all of the mice you enroll in your study are going to become diabetic. For every 10 female mice you place in your cohort, you would only expect 8 of them to be diabetic by 30 weeks of age. So if you need 10 diabetic females, you need to enroll at least 13 females at the start of your study. Second, disease onset is variable. Do you need all of your mice to become diabetic at the same time in order to start your experiment? You might consider adding mice into your study on a rolling basis or alter your treatment to match disease onset instead of specific age time points. And third, disease penetrance differs greatly between the sexes, with almost twice as many females becoming diabetic compared to males. Therefore, if you need both male and female mice for your study in equal cohorts, you will have to enroll twice as many males at the very beginning. So again, knowing the published characteristics of your strain prior to starting your experiment will be critical to ensure to plan your study. And if you're working with a strain with minimal publications, you might consider a pilot study to detail the phenotype progression in your own facilities. Another common model for diabetes research often models type 2 diabetes, a high-fat diet feeding plan to generate obese and sometimes diabetic mice. While this seems like common sense, if you feed a mouse a lot of fat in their diet, they should gain weight, it's not quite that simple. This is a great example of how sex differences are important to know, as well as strain differences before starting mice on a study. Several strains, such as the C57 Black 6J and AJ mouse models, when placed on a high-fat diet, have male mice that will gain significant amounts of weight. These mice may serve as models of obesity, prediabetes, or diabetes, depending on the strain. However, it's important to note that not all genetic backgrounds respond to high-fat diets. For instance, the FVB and SJL strains don't become obese when fed these diets. It's likely there are several underlying genetic factors contributing to these phenotype differences. In addition, male mice are the most commonly published upon high-fat diet mouse, but this isn't to say that female mice don't respond to a high-fat diet at all. Some female mice may respond, however, it doesn't seem to be as robustly, and it's much more variable than male mice. If you do need to use both sexes in your research, it will likely be important to perform these pilot studies in order to properly size your cohorts. Another consideration when planning your experiment is how you're collecting your data. Often in the case of diabetic models, blood glucose or insulin values are one of the experimental readouts. Blood can be obtained from several different sites: tail vein, retroorbital bleeds, facial veins, cardiac puncture, the list goes on. Many studies have been published, such as the one highlighted here, that compare the glucose and insulin values on blood taken from the same mice within a few moments of each other from different sites. Here, mice are sampled from the tail vein and by retroorbital bleed at the same time. The total time for both collections was under 4 minutes total, and you can see some pretty interesting differences. The paper goes on to resample mice a week later and analyze these results combined, but the ultimate conclusion is that on average, retroorbital bleeds have about 11% lower glucose values and a striking 40% higher insulin values compared to blood sampled from the tail vein. The important thing for your work will likely just be consistency. You can pick whichever route is best for you to obtain your samples, but being consistent within each cohort and over the course of your experiment will provide you the most accurate results. Similarly, the method used to measure blood glucose is going to be important through the course of your work. Glucometers, biochemical analyzers, glucose oxidase assays, choose whichever you're most comfortable using in your facilities. However, once you've made your choice, it's likely best to continue to use the same assay throughout the course of your experiment, and certainly between mice in the same experimental cohort. Shown here is some data collected at JAX to evaluate differences between a glucometer and a chemical analyzer. As you can see with some of the measurements, such as the one in the teal circle, they're very similar when run with both methods. But others, like the one pictured here, are vastly different. The glucometer reading is around 550 mg per deciliter, while the chemical analyzer has it about 450 mg per deciliter. Now, certainly, both of these readings are diabetic, but when you're looking to run analysis on these numbers, this could mean a really big difference. We do always want to mention that while keeping the method consistent is important, time of day is another important factor. Try to plan your experiments so that you're able to take samples at the same time of day, keeping in mind that mice are active during the dark cycle, so in the early morning, they've likely just eaten and may have higher blood glucose levels naturally. If you don't sample mice until the late afternoon, keep in mind that the mice have been sleeping all day and likely will have lower glucose values. Like all instruments, glucometers do need routine calibration. This is often overlooked in a lab setting, as many times there are multiple people in lab using the glucometer, or it's being left in the mouse facility and not thought of as often as it should be. Glucometers should all come with control solutions and instructions on how often to calibrate. We always recommend assigning someone in the lab to be the keeper of the glucometer to ensure this is happening on a regular basis, ensuring the results you are reading are accurate. In addition, glucometers tend to have limits for how much glucose they can detect. Many have a limit around 600 mg per deciliter, such as the one we've historically used at JAX. So what this often means is that any mice that have a glucose level higher than this will either just read high or 600 mg per deciliter, depending on the brand. You might be familiar with seeing results such as on this table, where the maximum reading we can obtain is 600 mg per deciliter. Now, this doesn't necessarily mean the mice didn't have a blood glucose over that amount, but that's the highest we can detect with this method. If you do need to detect blood glucose values higher than this, there are specific glucometers you can find that have extended detection limits. Again, it's important to know before you begin your study and purchase your equipment. One example of a study that was able to show a high blood glucose reading is below. As you can see, these investigators were comparing blood glucose values during the light and dark periods in the NOD mouse model, known to have high blood glucose levels. During the light on period in the open circles, we can see how their blood glucose values increase over time, reaching a maximum of around 600 mg per deciliter. However, the blood glucose values are much higher than this in the dark periods, as this is when mice are actively eating, and these numbers go as high as 800 mg per deciliter. With a standard glucometer that might max out around 600 mg per deciliter, you might miss this important trend towards the end of the experiment. Be sure to have an understanding of what you need for your experiment when choosing a glucometer. This might not be as critical if you know your mouse model will only ever have a maximum blood glucose level of 300 mg per deciliter. So moving into control selections, which are an important aspect of any experimental design. Often we think of wild-type mice or mice that don't carry the mutation as a good control. This could be a litter mate, which is a great way to control for genetic background of your model. Other times, such as when our breeding colony is homozygous breeding pairs, we don't produce any wild-type litter mate controls. So you might choose an inbred model or an F2 hybrid as a control based on the genetic background of your mutant strain. However, there will be some mutations where the heterozygous genotype might be an accepted control. This is really going to depend on your specific model and what your field accepts. One example of this is the OB mutation. Here we have the leptin OB mutation on the C57 black 6J genetic background. Homozygous mice are obese and diabetic. So this leaves you with several possible choices for controls. You could use littermate heterozygous mice or inbred C57 black 6J mice. Another example, along with a similar idea, is the leptin receptor DB mutation. The homozygous genotype is your experimental diabetic and obese mouse model. However, the heterozygous mouse exhibits a weight phenotype that's different from the wild-type mouse. It's not much, but it's clearly there. When you look at blood glucose and glucose tolerance, we don't see a difference in the heterozygous and wild-type mice. However, it might be important for your experiments to choose one of these genotypes over the other as your control group. It's important to know that this weight difference does exist, as some investigators tend to lump both heterozygous and wild-type mice in the same control group, which does lead to more variable data. Whichever control group you decide, we encourage you to be clear in your publications so other investigators can better understand your experimental design. In many circumstances, you may be looking for a control diet and not necessarily genotype. When working with diabetic mice that are diet-induced obesity models, a question we often hear is, what is the best control diet for my high-fat diet? There are several important factors to consider. The first being, what is your diet? High fat is a very general term and doesn't specify anything about the actual fat content. In the published literature, high fat can refer to diets with fat percentages anywhere from 20 to 70%, and most are referred to as high fat within the publication itself. Determining the exact diet that was used if you're trying to reproduce a study or performing a dose-response pilot study to determine which diet will work best for you will be the best way to determine which diet to use. Once you determine the high-fat diet, you'll likely have to pick your control diet. We always recommend contacting the vendor of your high-fat diet to determine if they have an appropriate control. Be sure to ask what the control diet is controlling for. Are they simply matching caloric content but switching the macronutrients? Are they using a purified diet? And if so, do they have a standard control? Often, a chow diet used within your animal facility will not be an appropriate control for a custom high-fat diet that you obtain. And again, when you publish your results, providing other investigators with as much information as possible will help reduce this challenge moving forward. Once you've chosen your model and designed your experiment, there is still another challenge to account for. Diabetic phenotypes are sensitive to environmental stress. We'll go through each of these in a bit more detail, but overall it's important to note that shipment of mice, housing considerations, and handling can all affect the phenotype of your model. Any mouse model of obesity that are caused by a custom diet, such as a high-fat diet, are likely to lose weight when they're stressed. This includes when these mice are shipped or moved between rooms in the same facility. JAX provides C57 black 6J mice that are started on a high-fat diet at 6 weeks of age and are available for shipment starting at 7 weeks of age. Once these mice are 12 weeks of age, we do provide a minimum body weight before we will ship them, shown in this table here. So this means all mice from the colony that ship at 12 weeks old must hit a minimum weight of 30 grams to be shipped. However, we're all aware that these mice will likely not eat during shipment. They will be stressed, which can lead to weight loss. This can be anywhere between 10 to 15% of their body weight, even in just a few days. When mice arrive, you may notice that the mouse that shipped at 30 grams now only weighs about 25 grams. This is to be expected. We recommend placing mice into their new cages, providing the same high-fat diet and access to water, and within 2 weeks, regain lost weight and continue gaining weight. I do want to point out that these numbers obviously increase as you order older mice. 26-week-old mice are weighed at JAX and will only ship if they're at least 47 grams. But this means they might arrive in your facility at about 40 grams. Please be aware of this. You likely do not want to use these mice in experiments for those first 2 weeks as they have lost significant weight. You may have to plan your mouse orders several weeks before you plan to perform your experiments to ensure the mice have gained back any lost weight during that acclimation period. For any mouse models, not just obese models, we do suggest keeping handling to an absolute minimum. Once these mice are in your facilities, weighing them often can also lead to continued stress, leading to a lack of weight gain. Once a week is the most often you should be handling these mice to maintain the obese body weight. Diabetic mice also require different practices than other mouse models. Group housing is always recommended in order to promote social interaction and assist these diabetic mice with thermoregulation, which can be disrupted by the disease. However, as many of these models do become obese or have increased urination, you may need to reduce cage density to comply with your facility's animal care regulations. This may also be necessary if you have any experiments, such as wound healing, that require lower cage density. Just as a quick reminder, never combine mice from separate cages into the same cage. Mice may become aggressive when they're combined with new mice, and if you receive mice in multiple cages from JAX, it's best practice to house them in the same configuration as they shipped to reduce this aggressive behavior. Bedding can be a critical difference between housing non-diabetic and diabetic mouse models. One common phenotype of diabetes is excessive urination, which in a mouse model will often require more frequent cage changes. You might also consider obtaining cellulose-based bedding, such as Alpha Dry, to keep the cages as dry as possible in between changes. I cannot stress this enough. If you are working with obese or diabetic models, please check with your animal care staff to make sure you do not have a corn cob-based bedding. love to eat corn cob bedding, and if you're working with a model that's supposed to gain weight from overeating, it's entirely possible they'll eat the bedding over their own food and not gain the expected weight due to the lack of nutrition in the bedding they're consuming. It's best to stay away from this type of bedding with diabetic models. We do always recommend providing enrichment to help decrease stress within the cages and promote natural behaviors. Every animal facility has different options. Simple nesting material, domes, or shelters, all of these are great options to help your mice. When it comes to actual food and water your mice receive, there are several things you'll want to pay attention to. First, always make sure your mice can reach the food and water. Some models that become obese no longer maintain the ability to stand on their hind legs and may not be able to reach food in the hopper or the water bottle. For these mice, you might want to provide a hydration bottle on the floor of the cage and be sure to place food pellets within reach. This might also be on the bottom of the cage. And speaking of pellets, if your mice are on a custom diet, it's critical to look into the vendor specifications for storage and spoilage warnings. Often high-fat food should only be provided for 2 weeks at a time, and during the cage change, all of the remaining food should be discarded and new food provided. Regarding water, so again, one common phenotype of diabetes is increased thirst, which means these mice will likely be drinking significantly more water than non-diabetic mice. So it's best to always make sure the mice have access to water. So one topic that we won't delve into too deeply, but I do want to mention, is the health status of your animal facility, because this can make a large difference in diabetes phenotype. The example that we're going to show here is a research colony of NOD mice started showing a reduced incidence in type 1 diabetes. Investigators ended up doing quite a bit of work and found that this correlated with the presence of segmented filamentous bacteria in their animal facility. By performing an antibiotic treatment followed by reintroduction of GI microbiota, rescued the diabetes phenotype, showing that because their environment had SFB, they were actually getting altered incidence of the diabetic phenotype. We put this one here to really highlight the differences that you might be seeing in your animal facilities, but there are many other examples in the published literature of diabetes phenotypes being very sensitive to different opportunists or pathogens that may be in your animal facility. In addition, we know that environmental stress can impact diabetes development on its own. The NOD mouse specifically is very sensitive to other forms of environmental stress as well. As we just mentioned, pathogen exposure might alter gut microbiome and lead to altered phenotypes. Because of this, we do always recommend maintaining these mice in the highest health status barrier your facility has to offer. One other example I'd like to point out using this model is environmental stress related to caging. Researchers actually found that female mice housed on the top shelf of the caging rack took longer to develop type 1 diabetes than those housed in the bottom of the rack. It's important to remember that mice are nocturnal and really like the dark. So mice housed on the top shelf are more directly exposed to room lighting than those housed on the bottom portion of the rack, which may lead to greater stress. Seemingly simple environmental factors, like where the mice are housed on the rack, may play a large role in disease progression. So today's presentation is to help you understand the challenges of working with diabetic mice, because once you understand these, this will allow for more consistent, reproducible results. Mainly, we want you to have a great understanding of what strain will best mimic your phenotype. Before you even start your study, it's important to realize not all models will recapitulate all diabetes phenotypes. When you're planning your experiment, each model will have different disease and sex penetrance, and maintaining consistent techniques for sample collection and readout will help give you the best results at the end. And sometimes to get those, you may need some pilot studies to best plan those larger experiments. Also, there are a lot of housing and handling considerations, especially when it comes to diabetic mouse models. We want you to follow some basic mouse husbandry tips to reduce colony stress, like a quiet room, stable light cycles, temperature and humidity, to reduce colony stress as much as possible, so you can contribute your experimental results to your treatment or experimental design and not perhaps from some erroneous environmental factor. In addition, there are special husbandry considerations for certain mouse models. Again, it's important to really have a good understanding of the model you've chosen before you begin your experiment. The Jackson Laboratory provides a wide range of services that may be able to help you plan and perform your studies. From providing mice to genome scanning and model generation, we're always happy to talk with you about your planned research. And thank you so much for watching this video. I hope this presentation has provided valuable information to help you achieve your research goals. If you have any questions about the information shared today, please contact us using the information on this slide. We would be delighted to support you with technical assistance.